Hello Year 9. Here is the feedback for the enzyme lesson from Monday of this week. So just a quick recap from previous lessons. We've been talking about enzymes as biological catalysts. So they are made by living things and they will speed up a reaction. So biological is from living things and a catalyst is something that will speed up a reaction. So this lesson looked a little bit more at how enzymes work. Talk specifically about the lock and key theory and explain why enzymes are specific, and a little bit about the collision theory, which is the kind of idea that for any reaction to happen there has to be some sort of collision between the particles to make that reaction happen. So throughout this presentation I asked you to write down some different definitions of things that you could then refer back to when you had questions. It was very, very clear to me that people are not writing this information down. Okay, so the first thing I asked you to write down were enzymes are biological catalysts made from proteins. They speed up chemical reactions. Okay, so again, someone had highlighted the word catalyst. They hadn't heard that before. So a catalyst is just something that will speed up a reaction without becoming involved in the reaction themselves. So you were asked to watch a video of an elephant's toothpaste reaction. This is a natural reaction. Hydrogen peroxide is a chemical and it will break down on its own into oxygen and water, but you can add different things to make the reaction happen faster, which is where the elephant's toothpaste bit comes in. So often we can use yeast. Yeast is a living type of cell and it has catalase inside it, but you could also use small pieces of liver or potato or different kinds of chemicals that will have the same effect, speed up that reaction. Okay, and then you were asked some questions about that reaction. Now I'm not sure if you didn't realize you can just flick back between slides, but it was quite clear that even though you'd watched a video and read that information, some people weren't really paying attention to what they were reading. So yeast contains an enzyme. The enzyme speeds up the breakdown of hydrogen peroxide into water and oxygen. So if you go back one page, it tells you third line down, hydrogen peroxide breaks down into oxygen and water. So you could just go back a slide and look that up if you didn't know. Enzymes are protein molecules that speed up chemical reactions. So you should have written that sentence on your piece of paper. Okay, why is this relevant to us? Well, all the chemical reactions in our body, what we call metabolism, the sum of all the reactions in our body, are usually all catalyzed by an enzyme. So I asked you to write this point on your paper. Enzymes are used throughout the whole body in processes like digestion and respiration. So a couple of people highlighted respiration. This is the chemical reaction that takes place in the mitochondria of the cells to release energy from glucose molecules. So putting all that together, the notes you've written and the recap, explain what an enzyme is and why they are important. Enzymes are biological catalysts. They are protein molecules that speed up chemical reactions in our body. That should have been written down on your piece of paper. How do they work? 2 key words that you were asked to write down. So the active site, a region on the enzyme that binds to a protein or other substance during a reaction. And the substrate. The substrate is a molecule upon which an enzyme acts. So the substrate is the thing that needs to be broken down or built up. The substrate bonds with the active site to form an enzyme substrate complex. Okay, so you were asked to write those 2 words down. And then there was a video that I made for you about the lock and key model. So there's 2 ideas about how enzymes work. One is called the lock and key model, where the substrate is like a key and the active site is like a lock. And if that key won't fit in the lock, then it's not going to work. But that's kind of been replaced now with something called the induced fit model. So you should have watched this video and the link will still be on the work, so you can watch it again if you didn't watch it the first time. I asked you to draw this diagram onto your sheet. This shows the induced fit model, but also a little bit of the lock and key model. Okay, so the substrate is the reactant in the chemical process, the thing that is reacting. The enzyme is the large protein with a unique sequence of amino acids. We know that proteins are made of amino acids. The active site is the area that attaches to the substrate. And if you look at the picture in the middle, you can see the substrate inside the active site, and this is the enzyme substrate complex. That's when they're locked in position on the enzyme. And then while they're locked in, the reaction happens, breaking those molecules apart. In this case, breaking them apart. They could also be connected together. And the thing that comes out then are the products. So the things that are produced are products. So if you drew this diagram, then these questions were super easy. What is the mechanism called where the substrate fits the specific shape of an enzyme's active site? It was called the lock and key. What is this part highlighted in red called? The active site. What is this part called? That was the substrate. What is this part? That was the product. And what is this light blue part? That was the enzyme. So if you label that diagram, then that should be perfectly fine. Now, we were thinking about enzymes not only having to match the active site, but have perfect conditions to work in. And I asked you if you could remember what pH was the measure of, and lots of you either had great memories or great Googling skills, but basically pH was how acid or alkali something is. Now, enzymes need the right temperature and the right pH, because if they're exposed to extremes of pH or the temperature gets too high, then the shape of their active site will change. And if this happens, the substrate won't fit into the enzyme anymore, which means the key won't fit the lock. And then when this happens, when the active site changes shape, we say that the enzyme has been denatured. It said, make a note of what this word means on your paper. So denatured means the shape of their active site may change. So what does denatured mean? If enzymes are exposed to extreme conditions, the shape of their active site will change. So enzymes need the right temperature and pH, and we use this word optimum. So a few people highlighted optimum. Optimum just means the best. So the best temperature or the best pH that it works out. And you can find this on a graph by looking at where the peak is. So mostly graphs will go up like this, and there will be a top point, and you draw a little line down, and that will tell you the best conditions for that enzyme. So you can see on the pH graph that at 0, 1, 14, at the extreme ends of pH, there's no enzyme activity. Temperature is usually a little bit different. It will still work at colder temperatures and just be a lot slower. But it will, at high temperatures, it will rapidly fall to 0. Okay, so here was another video, enzyme video, just to recap everything we've done. I hope you watched that video. And then there were some questions. So this graph shows the activity of an enzyme under different temperature conditions. What is the optimum temperature? So we're looking on the graph for the highest point. And if you look for that highest point and draw a dashed line down, you would see that the optimum was 35 degrees Celsius. Suggest what would happen after 35 degrees Celsius. So you can see that the enzyme activity rapidly falls towards 0. So the extreme temperature changes the shape of the active site, and this is where the enzyme is being denatured. Pepsin, amylase, and lipase are all enzymes, state their optimum. So on each of these graphs, we're looking for the top point. The dotted lines are drawn down. You just need to make sure you wrote the right enzyme with the right pH. So pepsin works best at pH 2, that is in acid conditions. Amylase works best at pH 7, which is neutral conditions. And lipase works best at pH 12, which is an alkali condition. Now, explaining the rate of these reactions. So I mentioned earlier something called the collision theory. And basically, whenever any reaction's happening, the reactants will start to collide. So, for example, if I put metal into acid, the metal particles will collide with the acid particles, and that is when a reaction will happen. Now, as you increase the temperature, the particles get more energy, which means they move around more and they collide more often. So the rate of reaction increases. If there's more collisions, then there's more of a reaction. So towards the end, it levels off. You can see in kind of section 4 on this graph, as the reactants are being used up, so there's less particles available to react, the reaction slows down a little bit. And this is what's known as the collision theory. When you get to year 11, there's an entire module about the collision theory, so you will learn a bit more about this. So, quick check from your notes that you made from the video and the diagram and things like that. What is the lock and key theory? Why was it called this? So, every enzyme has an active site with a unique shape that fits onto the substrate involved in the reaction. Enzymes are picky and only speed up specific reactions because for the enzyme to work, the substrate has to fit into the active site. If the substrate doesn't match the enzyme active site, then the reaction will not be sped up. This is like a key fitting into a lock. The key is the substrate and the active site is the lock, and if they don't fit, the lock won't work. It's the same thing. If the substrate won't fit inside the active site, then the reaction won't be sped up. What did denatured mean? An enzyme is denatured when the shape of the active site changes so the substrate can no longer fit in it. 2 ways an enzyme can become denatured, so high temperatures and extremes of pH. So by extremes, I mean like pH 1, pH 2, and then pH 13 and pH 14. However, as we saw from that graph with the 3 different enzymes, if you were best at pH 12, then an extreme for you could be 8, because that would be too low for you to work in. Now, happiness check. So far we've covered the organization of living things, digestive system, digestive enzymes, and how enzymes work. The organization part, everyone seemed fine with. No one highlighted that. But the digestive system, enzymes, all highlighted. But I would like to point out a few things. So please make sure you're watching the video feedback. Ironically, you are probably the person who is doing it, because if you're listening to this message, you obviously are watching the video. I get YouTube stats, so I know out of the class of 22 people doing the work, 4 people are watching the videos. So you can't say you haven't heard these words if you're not listening to all the work that is sent to you. I would also say that when you are asked to write and draw something on paper, you need to do it so that you can look back when questions come up. There was lots of things in the presentation today you were asked to copy down, and then there was questions straight after about it, and people hadn't written the notes down, which made it obvious because you couldn't answer the questions that were about the diagram you just did. And my final point is that email me when you get stuck. I think I've had 2 messages in 3 weeks, but there's obviously people who are more stuck and are not asking for any help when they get stuck in the work. So it's been a pleasure feeding back to you. I will see you after the next lesson, and I hope that I will hear from some of you if you get stuck on the next piece of work. See you later.